Hi guys, welcome back to another episode of Last Minute a channel that helps you with all your problems in life sciences. Even if it's the night before your exam or your test, or you are starting a new section in your life sciences and you're freaking out, this channel is here to help calm all those nerves down and make things easier and understandable for you. The subscribe button, it is free and you will not regret it. So make sure you guys leave a comment as well about maybe what you didn't understand and what would you like to see more from this channel in terms of topics, and other suggestions are also welcomed. But let's get into what we'll be doing today. Today we'll be looking at the cell cycle, breaking down mitosis, and also learning about cancer. So by the end of this lesson, we should be able to describe the cell cycle, explain the stages of mitosis, understand the role of mitosis, and also explain how cancer develops. Honestly, we will be looking at everything related to cancer, how it comes about, what causes it, and also the treatment. So let's just get to it immediately. So first thing first, we will be looking at the cell cycle. We have spoken, our last video, we've spoken about cells, how the organelles and the function of the organelles in the cell. Now we're looking at the cell's life cycle, basically. So first of all, in the cell life cycle, the longest phase is the interphase. During the interphase, the cell grows into its maximum size and it performs its normal cellular functions. It replicates DNA and prepares for cell division. Now, interphase is also divided into different stages. There's G1, G2, and the S phase. So during the G1 phase or the G1 stage of interphase, what happens? This is immediately after the cell has divided into two daughter cells. So basically, it's almost like the childhood phase of the newborn phase, toddler phase, and childhood phase of the cell. In this phase, the cell's main focus is to synthesize proteins and also increase in size. And it remains in this phase for a long time. Obviously, it's still growing and it's still developing and all of that. Then it enters into the S phase. The S phase is almost like puberty for the cell. So in this phase, what happens is DNA replication. The cell will make an identical copy of each of its chromosomes. So remember, a cell has 2 chromosomes, right? So they'll make a copy of each of them within their body. So these chromosomes are within the nucleus, and these chromosomes also carry genetic information of the cell. Like if the cell codes for hair color or light absorbing information and all of that, since all our cells in our body have different functions that keep us alive, then that genetic information will be in the cell and it will be duplicated, so then it will be passed on to new cells that will be made by this current cell. Then the cell enters into G2 phase. This happens after the cell has went through the DNA replication or duplicated phase. During this phase, the cell continues to grow and undergoes normal cellular functions, performing its functions in the body. And then towards the end of the G2 phase, what will happen is that the cell will now start replicating its organelles in preparation for mitosis. Okay, so like I mentioned, interphase is the longest phase of the cycle. It makes approximately 90% of the cell cycle or cell's life cycle, and then 10% is mitosis. So now let's get into the mitotic phase of the cell's life. So the meiotic phase is broken into two: mitosis and cytokinesis. So those are the two processes that are part of the meiotic phase of the cell's life. So mitosis is the division of the nucleus. So we just prefaced before that before the cell enters mitosis, it goes through, it does DNA replication. DNA replication occurs within the nucleus of the chromosome, where the DNA or genetic material is copied and doubled. And now the cell is already getting into mitosis. There's already double the amount of DNA in it that has two identical copies of the genetic information of the cell. Now the purpose of mitosis is to divide this into two different nucleuses. Two nucleuses will have the same or identical set of DNA or chromosomes. They'll have the same genetic information. So that's the purpose of mitosis. And then cytokinesis is the division of the cell itself. If you go back to our videos talking about the organelles, you'll understand that the nucleus is inside the cell. So go back to that if you didn't watch that video to understand the structure of the cell. So the cell in cytokinesis, as I was saying, is the division of the whole cell. The cell cytoplasm and the rest of the cell splits. The cell membrane pinches in and divides into two separate cells. So in mitosis, you will have one cell with two nucleuses. And at the end of mitosis, that cell will undergo cytokinesis, where its cell membrane and its cytoplasm will split, forming two separate cells, and each nucleus will go to one cell. So cytokinesis is the actual splitting of the cell into two bodies. I hope that was. Before we get into mitosis and all the phases, let's now look at the structure. Once you guys understand how the structure of the chromosome looks like, so that when I'm explaining each phase, it makes sense. So when you look at this image, the X structure here, this is a chromosome. And this X structure is a duplicated chromosome, meaning this is a chromosome that had underwent DNA replication. And now it has been duplicated. And the single line here, the single chromosome, is actually a chromatid. Single chromosome. So what this thing called the centromere is the thing that is holding two sister chromatids together, forming the X or forming the duplicated chromosome. So if a chromosome is a straight line, just one line, like single line like this, it's a single chromosome, it's a chromatid. But if it's an X, it's a duplicated chromosome, meaning it has underwent DNA replication. And this single line is a chromatid and the double line is a chromosome. And those squiggly lines inside the chromosomes is the genetic information. So when we start naming these structures, it will make sense for when we start looking at the mitosis phases and cytokinesis. So that is easy for you to remember how each phases of mitosis occur. So let's just basically name the phases. We have prophase, metaphase, anaphase, and telophase. And then the last phase is cytokinesis. So in order for you to remember the order, you can name them. You can, before you start your exam or your test, you can just write on your question paper IPMAT, like IPMAT or IPMAT, IPMAT. That is interphase, prophase, metaphase, anaphase, and telophase. So you can know the order. And also, going back to the chromosome structure, the chromosome or the body cells or somatic cells, which are your bodily cells, they have, like a human being in general, they have 46 chromosomes. 46 chromosomes are your somatic cells, or your body cells. 46 chromosomes, and half of those chromosomes come from your mother, and half of those, the other half comes from your father. So 23 comes from the mother, and 23 comes from the father. So your sex cells, like your egg cells and sperm, each carry 23 chromosomes. So sperm carry 23 chromosomes, and the egg carries 23 chromosomes. And when conception happens, when fertilization happens, the 23 from the sperm and the 23 from the egg fuse, and they make 46 chromosomes, and that's how a human cell is then made, and then the baby is made. So just putting that out there so that you guys can know. Somatic cells are your bodily cells, and there's 46 somatic cells, and they are all diploid. And then your sex cell is haploid, meaning it has 23 N, and diploid is 46. Let's start different phases in mitosis. So the first phase, it's the setup phase, it's prophase. We already spoken about interphase, now we're in prophase. Now cell division is now starting. So what would happen inside the cell? The DNA would condense into visible chromosomes. The chromosomes lay out properly, making sure that they are nice and visible. They shorten, thicken, and they become visible chromosomes. And then the nucleus disappears, and the nuclear membrane as well. The nuclear membrane and the nucleus disappears. Now this leaves the genetic information, or the DNA that was inside the nucleus, that is now had been replicated or duplicated. It's now sitting out in the open in the cytoplasm of the cell. And what else that happens is that there's this little dot that is called a centriole. Those centrioles, what they do is they start moving to the opposite poles of the cell, and they start to grow spindle fiber, little tentacles, like they start looking like spiders basically. That's what happens. They go to the north pole, the south pole of the cell, and they start growing little legs, or that are called spindle fiber, like they're spiders. They start looking like spiders. This happens in an animal cell, but in a plant cell there are no centrioles to move to the poles, so the spindle fiber is just randomly formed in the cytoplasm. So that's the prophase. Prophase, key sentences to make sure you have on your exams or your test, or to note: DNA condenses into visible chromosomes. The nucleus, or the nucleolus, disintegrates, or we can just say disappears. If it's a animal cell, if they mention in the question paper animal cell, then you say the centrioles separate and move towards opposite poles. If they mention that, so for an animal cell, the centrioles move to the opposite pole and form spindle fibers. But in a plant cell, spindle fibers forms in the new cytoplasm. Just leave it at that. Then we move on to the metaphase. Metaphase, it's the middle phase. Meta, middle. MM. Middle, meta, middle. Why? Because now what happens here is the chromosomes all line up in a straight line on the equator of the cell, in the middle of the cell. The equator, middle, metaphase. The chromosomes line up on the equator of the cell. The chromosomes appear in a straight line across the middle of the cell. Each chromosome is attached to a spindle fiber by its centromere. So what happens is that, remember the spiders that are forming at the opposite poles of the cell? They attach themselves to the centromere of each chromosome, or the chromosomes. And what happens is that then the chromosomes will line up on the equator of the cell, or in the middle of the cell. So spindle fiber attached to the centromere, chromosomes line up on the equator of the cell. Anaphase, apart. Anaphase, apart phase. So what happens is that now, remember what we say, chromosome is X. X with a dot in the middle of the X. The spindle fiber attached to the dot in the middle of the X. And if you remember, the X represents the chromosomes, and each straight line that forms the X is a chromatid. So what happens is the spindle fiber during the apart phase, the anaphase, when the spindle fiber starts to shorten, it's like the spider is pulling its victim in the web. When the spider starts shortening its legs, spindle fiber shorten, what happens? It pulls each spindle fiber. The other one is at the bottom, the other one is at the top. It's pulling. What happens? The X is broken, and now it's just one line is going the other way to the top, the other line is going at the bottom. So the chromatids are pulled to the opposite poles. So what will happen in anaphase? You get your first mark by saying the spindle fibers shorten, 1 mark. The chromatids, 1 mark, because you said chromatid, you're not saying chromosome, because chromosome is the whole X. That's not what happens. If you look at the diagrams, your X is now broken. The chromatids are pulled to the opposite poles of the cell. So now we move on to the next phase, the telophase. What happens in the telophase? Telophase 2. Telophase. Now we're going to have 2 new nuclei. What happens? A cell membrane reforms around the daughter chromosomes or the chromatids. The daughter chromosomes or the chromatids, a new nuclear membrane forms. So remember, we got one at the top, one at the bottom. So that means 2 nuclear membranes are forming that are now gathered at each pole. And then the cell membrane reforms and the 2 nuclei are formed, and the DNA chromosomes start to relax and turn back into tiny strands. So they recoil all back together again. So now you have 2 chromosomes in this one cell. So what happens in telophase? The nuclear membrane comes back, the nucleus is formed, and now we have 2 nuclei instead of 1. Cytokinesis. As you can see, a cleavage starts to form. The cytoplasm of the cell starts to divide, and that's what cytokinesis is. So cytokinesis is not a stage of mitosis. Well, after telophase has happened, mitosis is done, completed. Because we told you mitosis is the division of the nucleus. You have 2 nucleuses now. Now we have going through cytokinesis. Cytokinesis is the cytoplasm dividing and it's splitting into 2. So in the animal cell, you will see a cleavage forming. You can say that cleavage forming, or you can say infolding of the cytoplasm divides the cell into 2. But in a plant cell, a cross wall is formed by the cell plate, dividing the cytoplasm into 2. So in a plant cell, you'll see just a straight line forming in the middle. That's the cell plate dividing the 2 nucleuses. But for an animal cell, an infolding or a cleavage will divide the cytoplasm into 2. Now we have 2 identical daughter cells, which are identical to each other and to the parent cell. And now we're done. Now let's look at the role of mitosis. So mitosis is important for cell development. It's important for cell development and growth. So basically just is important for your whole body growing. Basically you're growing taller and you're growing hair, you're growing lashes, you're growing your nails. All of that is through the help of mitosis, because it helps increase number of cells in the body and enabling organisms to be more complex and multicellular, as we have 1,000 different, millions and billions of different types of cells in our whole body. We have skin cells, cells in our eyes, hair cells, all of those things. That's what mitosis help with, or it plays a role in. And also cell replacement. Again, your hair cells get damaged all the time, your skin cells get damaged all the time. Maybe you got cut, maybe you got sunburned. Mitosis helps to replace all of those damaged and hurt cells that you've lost, and making sure that they grow back, you continue functioning and living as you are. So now let's look at cancer. So cancer is a group of cells characterized by uncontrolled cell division, which leads to growth of abnormal tissues. So cancer cells grow and divide uncontrollably and form masses of cancer cells called a tumor. So a tumor is a mass of cancer cells. And so now let's look at the differences between normal cells and cancer cells. So cancer cells do not listen to signals to stop, the body signals to stop growing. So when your body is signaling to the cell to, okay, you've gone through cell division, it's time to stop, it doesn't listen. So cancer cells do not respond to signals from the body, it just keeps on dividing. And also cancer cells, as they grow, or the tumor as it grows, it releases proteins that attract new blood cells, stealing blood vessels from normal healthy cells, which is how your systems in your body end up dying and stop working. And cancer cells also have the ability to spread, or I could say some tumors have the ability to spread in the body by the blood system. And those type of tumors are considered malignant. That is like the dangerous. That's when you know, okay, you have cancer, you have to do something about it. But other tumors, they cannot spread to other tissues, and they are known as benign. So meaning they're not cancerous. So what will happen is that let's say you are checking your breast for lumps, as you should, and you feel there's a lump in your breast, and you then go to a doctor, book a doctor's appointment, you go to a doctor, and the doctor's like, okay, I can feel the lump. Or they will also do a mammogram, which is almost like an x-ray for your breast, and they will see that if they actually are able to identify the tumor, then they will book you a surgery. They will, and this is called like a biopsy. They'll remove this mass if it's possible, and then they'll conduct a biopsy. So it's removing that group of cells or that tumor, and then testing it to see if it is malignant or benign, and then from then onwards you'll get your treatment accordingly. So there are different types of cancers. There's breast cancer, lung cancer, and leukemia, which affects your blood. And some causes of cancer include smoking, cigarettes, radiation, x-rays, and UV lights. That's why it's important to wear sunscreen and don't smoke, and try to limit your exposure to radiation and stuff like that. So like maybe don't do laser all the time, and don't do x-rays unnecessarily unless you have to. Just stuff like that. Just be cautious and everything. So some of the common treatment for cancer includes radiation, chemotherapy, or surgery if it's possible and safe. So yeah, that's it for about cancer, and I hope it was clear for you guys to understand. And that's it for today. Thank you so much for watching. Thank you so much for tuning in and finding value in these videos. I appreciate all of you guys and all of your guys that are subscribing. If you watch this video and you are not a subscriber, please make sure you subscribe, because obviously you see that this is pure gold that you've stumbled upon. So guys, please subscribe, please like, please share these videos to your classmates, to your sibling who is still in school studying life sciences. So make sure you share these videos. There's plenty amount of A's to pass around, so make sure you pass them around. Be generous, be giving, be loving, and all of that will be repaid to you. And who knows, maybe we'll just have straight A's out of your report. So thank you so much for tuning in, and I hope you guys are having a great time. at school and are studying well. Good luck with your studies, good luck with your tests and exams, and God bless. See you on the next one. Bye.